My name is Anmarie Sica, and I'm going to be talking about cells in this video, but specifically the cell organelles, so the different things inside the organelle, or inside the cells that help it produce its job. So I'm going to go ahead and get jumping right into it. So the cells are made up of different things, like so the cell has a membrane, as you see here, and then which I guess acts as the barrier. And so in the membrane, there are these different things called the different organelles. And then the fluid around it, so the fluid in here is called the cytoplasm. But like I said, we're going to be talking about organelles. So I'm going to go ahead and jump right into that. So organelles have different structures that are different structures that carry out unique tasks or functions. And so what do I mean by that? Each organelle is different with its own function, but also is unique because it is according to what type of cell it is. So like, liver cells have a different job than muscular cells. And so they're actually also mostly located in the cytoplasm. So first we're going to talk about is the nucleus. Now the nucleus is usually the largest, so we'll go ahead and draw that. It's usually the largest, and it actually contains chromosomes, which are the genetic makeup of the organism, and also the genetic control center. So this one's actually unique in a way to most of the other organelles because it has a double membrane, which means that it just is double-lined, if you will. Now, the fluid that's inside the nucleus is called the nucleoplasm. So very similar to the cytoplasm, but they're not plasms. And so, okay, yeah. And then there is also what's called the endoplasmic reticulum. So this means basically little. Hey, what's up?
And we're back.
Sorry about that. So the endoplasmic reticulum basically just means like little network of cytoplasm, or little network in the cytoplasm. I'm sorry. So this is like a system of cisterns, and there are two different types of endoplasmic reticulum. So we're just to keep it easy, it's going to be rough ER and smooth ER. So, yeah, and so I'm going to go ahead and actually draw real quick the rough ER. And so rough ER, the endoplasmic reticulum is generally really close to the nucleus. So, I'll go ahead and draw it like this. It's like these resemble the different cisterns. And so, yeah, and so what makes it rough, basically, on top, what makes it rough, what would make it rough is specifically these little things, these little pockets of protein called ribosomes that are attached to it. So it just gives it the appearance that it's rough. So I'll just bring it up closer to you. So yeah, if you can see the red marks there. And so they are generally parallel and flat, the cisterns, and then they have the ribosomes on top. And it usually runs along the nuclear envelope or the nuclear membrane. And so yeah, most of the proteins that are produced here are actually secreted out of the cell or are sent to another organelle. The smooth ER are these more like tubular systems. So right here, I will go ahead and draw these little tubes here. That'll be like the tubes. And so the smooth ER does not have ribosomes, and they are mostly in cells that detox, like the liver, the kidneys, cells, and they are also in cells that synthesize steroids, such as like the testes or the ovaries. And so they are also extensive in skeletal and cardiac muscles, because they store and release calcium. There are 3 ways that you can, those are basically the ways that you can differentiate smooth and rough ER. The smooth ER also, like I said, it produces steroids. It also produces lipids. It detoxes the cells, and then manufactures basically all membrane, like the whole membrane in the cell. So with the proteins that they use. And so next, since we're talking about the ribosomes, or since we're talking about the endoplasmic reticulum, the rough ER has these little things called ribosomes, and so that's going to be considered another organelle. So ribosomes are these little small pockets of protein and RNA, actually. So they are found not just on the endoplasmic reticulum or the rough ER, but they're also found in the mitochondria and the nucleoli, actually. So inside the nucleus, there are little pockets of protein, or ribosomes. And so they are attached to, basically their function is to assemble amino acids to be able to make proteins and that are specified by the genetic code. And so they also so that would mean that they also read the genetic code, or the mRNA, the messenger RNA. So yes. And so next, actually the next organelle that we're going to talk about is called the Golgi complex, or the Golgi apparatus. And so the Golgi complex actually is also another system of cisterns. So, except they look more like, excuse me, like so, sorry. They look more like this. And so, they are, basically what they do is they receive the proteins from the rough ER, and they sort them, and then they add carbs to them to be able to add the finishing touches before they are sent off to other parts of the cell. And so they almost look like, to me, they almost look like a stack of pita bread. So that's really helpful. And then they also consist of almost attached to the ends of the cisterns are these things called Golgi vesicles. Or Golgi vesicles. And so, which sometimes actually becomes what we're going to be talking about next, which is the lysosomes. So Golgi vesicles sometimes become the lysosome. And so lysosomes are like these little basically they package the enzyme, they are little packages of enzymes that are bounded by a membrane. So that are basically right there, around the ends of the endoplasmic, and the Golgi complex. So they are variable in shape, because they don't have a standard structure. And basically what they do is they hydrolyze substances such as proteins and carbs. In the liver, They're really prevalent in the liver, so they help break down glycogen and release the glucose into the bloodstream. In the blood, white blood cells, they digest the bacteria, and then they also dispose of components of, like, almost like that are surplus or that are extra, and then they turn them into nutrients, so that way they can be used differently, used up in the cell. And so they also actually aid in the process of something called cell suicide. So suicide, or cell suicide, is basically like some cells are just meant to do one job once and that's it. So the lysosomes help it to die. Otherwise, it would be like a buildup of useless cells. And so, yeah. The next thing we're going to talk about is the peroxisomes. So the peroxisomes, hold on one second. So the peroxisomes are, sorry, I got sidetracked. So the peroxisomes are similar to the lysosomes. So we'll just draw them around here. Except they are made up of different enzymes. They are produced by already existing peroxisomes, actually. And this is kind of like a joint effort between the endoplasmic reticulum and the mitochondria, which we'll talk about that next. But the functions of the peroxisomes, they they oxidize organic molecules. They neutralize, if you will, free radicals. They help detox, and they decompose fatty acids for the mitochondria to use to make ATP. So these are abundant in liver and kidney cells as well. And so the mitochondria is what we're going to talk about next. The mitochondria, this is considered the powerhouse of the cell. I thought it would be that the nucleus was going to be considered the powerhouse of the cell, but actually the mitochondria is. And so the mitochondria, we'll go ahead and draw it like this, is also actually double-lined, just like the nucleus, or with a double membrane. And so they are also made up of these things that are almost like shelves, or that like fold out of the membrane. And so that is actually called the cristae. And so, yeah. And then the inside of and I keep breaking the cell. So the inside, or the fluid inside, is called the matrix inside the mitochondria. And so the mitochondria also has ribosomes in it, interestingly enough. So yeah. And then they generally vary in shape, but they're most of the time, I guess, or from what I've seen, they look more like a bean. So yeah, sometimes drawn into one. Sometimes two actually join into one, or they divide into two, which is really interesting and unique to the mitochondria. And yeah, so they have. So this is actually where energy is not made here, but it is where they extract the energy from organic compounds to make ATP. And so the last one that we're going to go ahead and talk about is called centriole. And so the centrioles are short little cylinders of tubes. So I'm just going to look at it, or I'm just going to draw it as if we're looking at it from the top. So it's like almost like we're looking down. And so these actually only work in mitosis or in cell division. And so I'm actually going to go ahead and talk about that in the next video.